Think back a little earlier, we introduced two types of cells in the nervous system. The neurons, which we've been through with the axons and the dendrites, they conduct electrical activity or electrical impulses, action potentials. We had a lot of names for it. Those are the information carrying cells, the neurons of the nervous system. But we also said there's also the glue cells, the glial cells, also called neuroglia, which literally means nerve glue. So these cells, we said earlier, are the supportive cells or the supporting cells of the nervous systems. They cannot conduct impulses, but they do everything else, right? They support the neurons, they nourish the neurons, they wrap around them. We saw Schwann cells for myelin. That's an example of a neuroglia or a glial cell. So the next part of this PowerPoint is to go through the 6 glial cells or neuroglia, and your job is to determine which ones are in the CNS, which ones are in the PNS, and the function of each one. So the next few slides will go through the 6 types of glial cells. So in each of these slides, I highlighted or underlined or bold typed the key words or the important words that will bring you to the answers. So first type of neuroglia is an astrocyte, which means a star cell. So it's shaped like a star, and you can see this one kind of looks like a star. The astrocyte is found in the CNS. So that's the first thing to know. Astrocyte is in the central nervous system. It's there for support, and it forms a blood-brain barrier. We haven't really described this yet. That's more AMP2, blood-brain barrier. But what it is, in the brain and right in the CNS, we have these tiny little microscopic blood vessels called capillaries. Actually, they're all over the body. But in the brain, the astrocytes wrap around them. This is so material can't get to the brain tissue very easily. Capillaries are blood vessels that can easily exchange fluid or material with the tissues. So material can go through the walls because these blood vessels are porous. It's not showing it here, but we don't want the brain tissue to be exposed to a lot of toxins or other chemicals that might be in the blood. So your astrocytes form this barrier, which makes it very difficult for chemicals to leave the blood and enter the brain tissue. It's just an extra level of protection in the brain called the blood-brain barrier, which is accomplished by the astrocytes. Your second glial cell are microglial cells. So number one thing to know, they're in the CNS, central nervous system, and the key word is phagocytized, or they're phagocytes. So they're patrolling your brain tissue and spinal cord tissue, and if they come into some type of, or come across some type of microorganism or just some debris, they can engulf it. And we know what phagocytosis is. It's when cells take in material from outside. The microglial cells are the phagocytes in your central nervous system. Next, neuroglia are ependymal cells. So first thing, know where they're found in the central nervous system, in the CNS. And what they do is going to be important in Unit 4. I know we're not there yet, so I'll just give a brief explanation. Cilia, well, they contain cilia, which we know what cilia is, those hair-like extensions that sweep material along. The cilia beat to circulate something called cerebrospinal fluid, CSF for short. What CSF is fluid that surrounds your brain and spinal cord. So we want this fluid to circulate right around your brain and around your spinal cord. And the way it moves around, how this fluid circulates around, is through the cilia on these ependymal cells. And it lines the central cavities of the brain. By the way, these brain cavities is where the cerebrospinal fluid comes from. So we'll have a lot of ependymal cells that will sweep the fluid along the central nervous system. All right, and now we get to the oligodendrocytes, also in the central nervous system, or CNS. So all of the neuroglia so far have been in the central nervous system. So all first 4 that we've been through are central nervous system. So the oligodendrocytes are the cells, here it is right here, that wrap around the axons in the brain and spinal cord, in the CNS. We saw Schwann cells before that wrap around axons to myelinate them, to produce white matter. That's not the central nervous system. That's in your nerves, right? That's your peripheral nervous system. In your central nervous system, one oligodendrocyte will myelinate many axons. You remember in the PNS, the myelin is accomplished by Schwann cells, which you need many Schwann cells. Remember the nodes in between the Schwann cells? Well, that doesn't happen in the in the central nervous system. The oligodendrocytes just form this continuous myelin over axons, and that's only in the central nervous system. This last slide on glial cells, or neuroglia, contain the two neuroglia that exist in the peripheral nervous system. They both start with the letter S, so you might want to remember that. The two that begin with the letter S are found in the peripheral nervous system, or PNS. The satellite cells, and you can see kind of wrapped around the cell body, we have the satellite cells, so they're surrounding a neuron cell body, and they just help regulate the chemical environment. Not a whole lot to say about the satellite cells, but what we've seen before, these tiny little cells that wrap around the axons, we've got many of them there. They produce the myelin around the axons in the peripheral nervous system. Those are the Schwann cells. And Schwann cells make these myelin sheaths. That's the white matter in the peripheral nervous system. Remember, oligodendrocytes produce the myelin in the CNS, but Schwann cells myelinate the axons in the PNS, or peripheral nervous system. A little more about the myelin. The myelin sheath is the Schwann cell, right? Myelin sheaths are Schwann cells around or circling around the axon. So we're looking right here at a myelinated fiber. Remember, a fiber is another way to say an axon here. So what we've never brought up yet is, why do we have myelin? Why do some axons contain myelin like this one does, where other axons in your body, in your nervous system, do not, which we haven't really seen a picture yet of an unmyelinated axon, but they exist. So here's why we would have myelin on a given axon. It says protects and electrically insulates an axon. So here's one axon in one neuron, but remember, in a nerve, you have thousands of these all bunched together in one cord, right? So information flows in the form of electrical impulses. Well, if there was another axon right beside it, we don't want the signals to get crossed, right, to interfere with other axons which are lying right beside it. So think of myelin as like insulation around a wire, right? So if you ever open up a wire and there's all these other little wires inside of it, and they have plastic around it that you can splice, and there's another little wire inside of that, well, it's kind of the same thing. So myelin helps insulate this axon so the electrical impulses don't interfere with other electrical impulses from other axons. But another extremely important reason why axons are myelinated is because what I have underlined here increases the speed of the nerve impulses. So if you actually were to time how quickly information flows down this axon here, it'd be much faster than if an axon was unmyelinated, because non-myelinated fibers conduct impulses, and that's information flowing in the form of electrical activity. That's what we mean by impulses. So a non-myelinated fiber will conduct these impulses much slower compared to a myelinated axon. Take, for example, let's say you put your hand on a hot stove. Accidentally, of course, right? So what's the first thing that happens? Well, your hand comes off right away, right? So that's a reflex because of that extreme heat that the receptors in your dermis are picking up. So those axons, that sensory integration center and motor output to your muscles to remove your hand from the stove, those are myelinated. We need speed to protect you, right, from damage from that hot stove. But then right after you touch that hot stove and your hand comes off, what do you feel like a second later or a couple seconds later? Is it starts stinging because it burned a little bit. So you don't actually feel that stinging for a few more split seconds. Well, That's information getting to your brain. Your brain's processing it. It's saying, well, that was hot. That takes a little more time for you to just kind of understand what just happened, a few more split seconds. So those are unmyelinated fibers, and you can see the speed difference between your hand coming off and then actually feeling that sting, that burn after, difference between myelinated and unmyelinated speed of impulse. And this is just a slide to show you that a myelin sheath is actually a cell, right? So if we actually look at one myelin sheath, we can see that there's the nucleus of the cell. So this Schwann cell is growing, and as it's growing, you can see the arrows. It starts wrapping around the axon, and it wraps more and it wraps more, and you can see the nucleus kind of being squeezed out at that leading edge, and it kind of just wraps around an axon like a little jelly roll. That's what a myelin sheath is, and that's what a Schwann cell is.